And it is talking about the introduction to genetics, chapter 11, the work of Gregor Mendel. We want to describe how Gregor Mendel studied inheritance. We want to summarize Mendel's conclusions about inheritance, explain the principle of dominance, and describe what happens during segregation. So, Gregor Mendel was a Augustinian monk, which means that he worked in a monastery. And the reason why he became one was because he was orphaned when he was younger in his life, and he was very inquisitive and he wanted to learn about science. So he figured that the best way for him to get an education and to be able to do what he wanted was to stay in the monastery. So he was very good in math. He was very good in astronomy, all kinds of different things. Very interesting. There's a video that I'm going to put the on Moodle that you can watch that goes with it. Heredity is what is passed on from one generation to the next. So genetics is a scientific study of heredity. Instead of looking at things like other people had previously and kind of theorizing about things, he actually did experiments. So that's why we call him the kind of the father of genetics. Gregor Mendel's experiments laid the foundation for genetics. He performed his experiments using garden peas. Garden peas are very easy to work with because you can easily control how they breed, or we call this process pollination. So the question is, how are heredity and genetics related to each other? Well, genetics is the scientific study of heredity. So why did Mendel use pea plants for his experiment, as it asked on number 7? Well, because they were easy to work with in terms of pollination. So this shows a flower, a pea plant, and many people call pea plants the perfect plant for doing genetic experiments because they have both male and female parts. Now, Mendel was very careful to establish what he called true breeding pea plants lines. So, for example, if they had purple flowers, then they had for many generations only produced purple flowers. Or if they're white flowers, they produce only white flowers. So we call them purebreds. Now this was important, and the reason why was because if you didn't do this, then he wouldn't know for sure that his experiments were successful. They wouldn't be controlled. Then he controlled how the pea plants were fertilized. He developed cross-pollination. So what that means is when you take the pollen from one plant, for example, this white pea plant, he transferred using a little paintbrush to the carpel of another pea plant. That's called cross-pollination. So the question is, what are true breeding plants and why are true breeding plants, pea plants, important in Mendel's experiments? Because it allowed him to control his experiments properly. What did what were Mendel's 4 steps for cross-pollination? Well, first he removed the stamen from the purple flower plants. So he took off the male parts that produce the pollen. Then he transferred from another plant, for example, in this case, a white uh, flower, to the carpel of the purple flower. So that's where we get the cross pollination. Then he allowed the carpels to mature into pods. And then after that, he took the peas and he uh, planted them to see what their offspring are. Now notice that they were all purple, even though he had crossed it with a white. Well, you might guess that the purple plant was dominant, or the purple flower was dominant to the recessive white. Now, traits um, are specific characteristics of uh, of a plant. Genes are chemical factors that determine the traits. Alleles are just different forms of a trait. So if you look at, for example, the yellow pea is that's a trait, okay, is a yellow trait. It's controlled by a by a gene that's a chemical factor, and it's the DNA. He didn't really know about DNA, and he didn't know about genes, but he did know that there was something that was creating that gene, that, that trait. And so it happens to be a protein that makes them yellow or green. And then he, an allele is basically just a different form of a trait. So there's an allele for yellow color and one for green. So alleles are represented by either uppercase or lowercase letters. The uppercase is the one that is um, selected, or the dominant trait is the one that's selected as being the letter that we use. For example, if there's the green pod happens to be the dominant trait over the yellow pod. So there would be a big G would be a would be the dominant, and the little g would be the yellow. Mendel tested a number of traits, including the following: seed shape, whether it was uh, whether it was wrinkled or whether it was smooth; seed color, whether it was yellow or green; seed coat color, on the outside of the seed there's a coat; pod shape, which is whether it is smooth or whether it is constricted; pod color, whether it's green or yellow; flower position, whether it's at the end or whether it's on the sides. We call that terminal or axial; and how tall it is, whether it's short or tall. So the question is asked, um, how are genes, traits, and alleles related to each other? An allele is a different form of a trait, and a gene is a chemical factor that determines a trait. So this shows the experiment between his first experiment. He took a tall plant and he crossed with a short plant. And notice that all of them came out tall. What did he call? These are called hybrids, the offspring of a cross between parents of different traits. The P generation is what he called the parents, the parental generation. The offspring were the F1 generation, or the first familial traits. So what were the results of the F1 cross? They were 100% tall. This was a surprise to him, because the fact of it was that um, they he expected there to be a blend between the two, as many other people thought. So the next cross was between the offspring from the first generation. So they call this the F1 cross. Notice that they were two tall plants from the F1 generation. The offspring were all were the offspring were there was 3/4 of them were tall and one of them short. Notice that the big T and little t always makes tall because of their the fact they're recessive. Now, how did he explain this? He took the purebred tall plants and the purebred short plants, and he put them together, and they ended up, he thought that they were segregating during the process uh, prior to the making of the fertilizing. Shh. So the when you put them together during fertilization, you get a big T and a little t. So these were 100% tall. Even though they had different alleles, they were 100% tall. It works. So he theorized that alleles were separated during segregation. Segregation takes place during gamete formation. Gametes are sex cells. There's pollen is from male, ovule is a female. He also theorized that alleles were united during fertilization. From this first experiment, he concluded that tall traits was dominant to recessive traits. So the question is asked, so what is segregation? It's the separation of alleles prior to fertilization. What happens to alleles during segregation? They're separated from each other. What are gametes? The gametes are the sex cells. They're the pollen for the males, for the male plant, and the ovule for the female. So what is fertilization? It's basically when the alleles are united during the process of sexual reproduction. Since 1/4 of the offspring from the F1 cross were short, Mendel concluded that the short trait had not disappeared, but it had been masked. This is an important question because previously they believed that there was a blending. Now we knew that it wasn't a blending. We knew that there were separate traits. He called them factors. We call this the principle of dominance. Alleles that mask other alleles are called dominant. Alleles that can be masked are called recessive. So the question is asked, what are dominant and recessive alleles? Dominant alleles make a product. Recessive alleles don't make a product. Okay, so dominant alleles can be masked or mask the other things, whereas recessive alleles don't.